The Hunger Games by Sam Polevoy, Jeremy Ansdell, and Rosa Park. Each year, an estimated 1 billion pounds of pesticides are applied to U.S. farms, forests, lawns, and golf courses. Humans face pesticide exposure through food and water residue. For example, herbicide atrazine is found in 94% of U.S. drinking water. In order to test this and find a potential genetic mutation that helps us to survive against these harmful pesticides, we will be using an animal called C. elegans for short to test for possible genetic mutations that help them to survive, and if these genetic mutations can also be found in humans. Ultimately, we're hoping to trace back genetic mutations to humans in order to find a way to help us better combat the harmful effects of pesticides. So you're probably wondering, what is a C. elegans? C. elegans are small round worms that are approximately 1 millimeter in length. They live in soil, which makes them a great candidate for our studies under pesticides. Their average lifespan is 2 to 3 weeks, making them a good model organism. And they share about 20 to 30% of genes with humans as homologs, so meaning genes that have similar functions. So let's get into the actual procedure of our project. First, we started by growing 40 to 50 strains of pre-genetically modified C. elegans. We then put them into one super plate for them to grow and reproduce. After the worms grew and reproduced in a similar habitat, we would wash them into 3 separate pesticide plates. Once they were on these 3 pesticide plates, they grew until starvation, meaning that they were in combat for the food that was left, i.e. the title, The Hunger Games. After the worms starved and were beginning to die off, we would transfer them to future generations and concentrations of pesticide plates. This would allow us to see which worms were surviving the best under each condition. After 3 generations of transferring the worms over, we would then wash the worms off the plates into test tubes to be DNA sequenced later. Once we had the DNA of the worms in the test tubes, we took them to the University of Washington and used these things called MIP probes to sequence the DNA and to see which genetic mutations the worms contained that survived the best under the pesticides. During the DNA sequencing process, we used probes to run over the DNA strand of C. elegans to see and look for specific strain markers that would tell us that the strain is contained in the final petri dish of C. elegans. So in doing this, it would run over the strand of C. elegans DNA until it found that specific marker, and that would send a ping to the machine, the DNA sequencing machine, letting it know that the strain was there. Ultimately, after it was done running through, it would generate a graph for us showing us the percentage of C. elegans contained on the plate. For example, in the first graph, it shows that there are less control strains than the mutated ones in the pesticides. In the second graph, it shows that the control strains grew very well, while the pesticide plates did not. In the third graph, the pesticide plates 2, i.e. P2, grew very well, while P3 did not. And in the fourth graph, none of the strains grew very well. The 0 point is the average for the growth of the C. elegans, so if it is above 0 on the graph, then it grew well, and if it is below 0, it grew poorly. So, we can use these graphs to draw several conclusions. First, there is a strong relationship between the pesticides tested and the survival rate of specific C. elegans mutated strains. For future work in the study, we hope to isolate specific mutations in order to reveal exactly which one influenced the worm's survival in their pesticide environment, meaning to find a specific mutation that helps the worms to survive better. And ultimately, this study has the potential to connect homologs between the worms and humans to help improve our understanding of how the chemicals influence the overall health of both species, and potentially find a way to help us better combat the harsh effects of pesticides.